And now we are going to have the morning session to start. The morning session is about life science creating paradigm shifts. Now I would like to introduce with you about Professor Dennis Low as this session's convener. He is the Associate Dean Research, Faculty of Medicine, Director of the Li Ka-shing Institute of Health Sciences, and Chairman of the Department of Chemical Pathology, the Chinese University of Hong Kong. He is also the Foreign Associate, U.S. National Academy of Sciences, Fellowship of the Royal Society, President and the founding member of the Hong Kong Academy of Sciences, the Future Science Prize 2016 Life Science Prize Laureate. Let's give a warm welcome to Professor Dennis Low.
Good morning. I'm Dennis Low from the Hong Kong Academy of Sciences and the Chinese University of Hong Kong. It's my great honor to be the chair for today's first session on life science entitled Creating Paradigm Shifts. Life science is one of the most rapidly developing scientific discipline of the 21st century. Advances in life science hold promise to positively impact human health, creating novel diagnostic approaches, and giving us new drugs and vaccines. And we have three distinguished speakers for this session. Even though the areas that they work in are different, the careers share certain similarities. First, each of them has made discoveries or inventions that have changed the world, hence befitting the title, Creating Paradigm Shifts. Second, apart from being world-leading scientists, each of our three speakers are also successful entrepreneurs. In this regard, one can perhaps see that one way to ensure that one's work is translated to real-world applications is to be closely involved in the process of commercialization. Our first speaker is Sir Gregory Winter, who did his groundbreaking work at the Medical Research Council Laboratory of Molecular Biology in Cambridge, UK. He invented a technology called phage display that greatly accelerated the generation of monoclonal antibodies, which have become a highly valuable class of drugs. And for this work, he was awarded the Nobel Prize in Chemistry in 2018. And Sir Gregory's talk today will focus on antibodies and antibody mimics. Our second speaker is Sir Shankar Balasubramanian, who is the Herschel Smith Professor of Medicinal Chemistry at the University of Cambridge. Sir Shankar is a co-inventor of the sequencing by synthesis approach, which has been commercialized by Illumina. This sequencing approach is amazingly 1 million times faster than the previous generation called the Sanger sequencing, and hence creating a revolution in genomics. And for this work, he was awarded the Breakthrough Prize in Life Science in 2022. His talk today is titled DNA, What's It Trying to Tell Us? Our third speaker is Professor Tak Mak, who works at both the University of Toronto and the University of Hong Kong. His best-known work is his cloning of the beta chain of the T-cell receptor. He has also made fundamental discoveries underpinning one of the approaches for cancer immunotherapy. In recognition of his work, Professor Mak has won numerous awards, including the Canadian Gardner International Award. And today, he will tell us something about the future of anti-cancer treatments. Without further ado, let's start the session. Thank you.
Thank you so much, Professor Low, for your exciting introduction. And we are thrilled to have our first keynote speaker, Professor Gregory Winter. He's the Fellow Trinity College, Cambridge, Emeritus MLC Laboratory of Molecular Biology from Cambridge, UK, Fellow of the Royal Society and Nobel Prize Laureate in Chemistry 2018. We are so expecting to listen to the topic, Antibodies and Antibody Mimics. Let's welcome Professor Gregory Winter.
Well, good morning, Ladies and gentlemen, Dennis, thank you very much for the invitation. And I'd like to thank the people here and, in fact, in general in Hong Kong for making me feel so welcome on this occasion and also whenever I come to this place. So I've taken the chances of putting my talk electronically in front of me, and I hope I don't lose it at some point by moving my finger too fast, but forgive me if I do. So what I'm going to talk about, if the slide is up, which it is work which was performed at the Medical Research Council Laboratory of Molecular Biology and two companies, Cambridge Antibody Technology and Bicycle Therapeutics, that I helped to found. And it involved many scientists working with me and independently. If you're interested, there's much more detail of the work to be found in the two web references. That's the Nobel Prize website and also Bicycle Therapeutics website. And I'm going to be talking about the development of antibodies as medicines over the last 30 years and our current efforts to develop small antibody mimics. So going back a long time, the earliest medicines were extracted from plants. And by the 19th century, chemists had discovered that many were organic compounds and that their medicinal properties could be altered by simple chemical modification. By the 20th century, the chemists were synthesizing entirely new compounds for use in medicines. In turn, this led to the growth of the pharmaceutical industry based largely on chemical drugs, at least until recently. But there's another class of pharmaceuticals, that's biologics, that have been transforming medicines and the pharmaceutical industry since the advent of recombinant DNA technology in the 1970s. These comprise proteins or nucleic acids or both, and come in a variety of forms, including antibodies, viruses, cells, or even RNA in lipid encapsulated nanoparticles. All, in my view, are biologics. So this slide shows the top 10 selling pharmaceutical drugs for 2023 based on sales predictions. On the left-hand side, antibody drugs are marked in red, chemical drugs in blue, and vaccines in black. On the right-hand side, the drug targets are in pink and the disease indications are in green. And there's a key at the bottom if you're really interested in any particular disease. Astonishingly, 7 of the top 10 selling pharmaceuticals worldwide are biologics, and 5 of these are recombinant antibodies. At number 1, with sales of $24 billion, is Keytruda, a checkpoint inhibitor antibody that's transforming the treatment of cancer. At number 3, with sales of nearly $13 billion, is Humira, an antibody for treatment of immune inflammatory diseases such as rheumatoid arthritis. At number 2 and 9 are the mRNA nanoparticle-based SARS-CoV-2 vaccines. And I think we'll see much more of nanoparticles and cells in the future of medicines, but currently, antibodies are the paramount biologic. Now, in many ways, the ascendancy of antibodies isn't surprising. Antibodies are nature's own pharmaceuticals, created to order by the immune system against agents of infectious disease. As shown on the right-hand side, IgG antibodies are large 150 kilodalton Y-shaped proteins with 4 chains, that's 2 heavy chains marked in red and 2 light chains marked in white. At the tips of the arms of the Y, the heavy and light chains come together through the variable domains, creating a binding site for the antigen. Now, binding alone may be sufficient to confer a useful therapeutic activity. For example, the binding of antibodies to a virus may be sufficient to block its attachment to an internalizing receptor and to block infection. But antibodies also have therapeutic activities built into the stem of the antibody. One such example is a binding site for a recycling receptor. Typically, proteins are cleared continuously by pinocytosis into endothelial cells and digested in their lysosomes. But antibodies are rescued by the recycling receptor, and that returns them to the cell surface, greatly extending their half-life in serum. The stem also has binding sites for receptors for immune cells and serum receptors, which can mediate the killing of pathogens, of which more in a minute. Now, the ability of antibodies to block pathogens, to maintain circulation for long periods of time in the serum, and to kill pathogens accounts for their ability in combating infectious disease. But in fact, it's their application to non-infectious diseases, such as cancer and immune inflammatory disorders, that has made antibodies the paramount biologic, not in its natural mode as an anti-infective, but in treatment of these other non-infectious diseases. In fact, in this case, the greatest utility of antibodies has been in the sustained blockade of cell receptors and their protein ligands. For example, tumor cells sometimes take advantage of natural mechanisms that stop cytotoxic T cells running amok. One such mechanism is the expression of a protein ligand that engages with the PD-1 receptor of cytotoxic T cells and switches those cells off. But an antibody such as Keytruda can block this interaction and thereby activate the cytotoxic T cells in tumors, leading to cell killing. By contrast, some inflammatory disorders such as rheumatoid arthritis and Crohn's disease are triggered by the binding of the protein TNF to its cellular receptor. By blocking the interactions with an antibody such as Humira, it's possible to block the inflammatory cascade and dampen down the disease. Now, notwithstanding the importance of sustained blocking, the killing activities of the antibody can also be important in treatment of non-infectious disease. Antibodies, for example, can kill tumor cells, which in this slide I've marked in dark blue in the slide, in multiple ways. For example, they can recruit the effector functions of macrophages, marked in pink, NK cells, marked in light blue, and complement C1q, which is in yellow. More specifically, macrophages, that's the pink ones, engage with IgG through their high affinity Fc gamma R1 receptors, leading to internalization and digestion of the target. By contrast, NK cells, that's light blue, engage with IgG through the Fc gamma R3 receptors, unleashing perforins that generate pores in the membranes of the target cell, leading to cell death. And serum complement C1q, yellow, engages with the matrix of bound IgG, leading to complement activation, osmolysis, and death. Now, these sets of effector functions, ADCP, ADCC, and complement, can all be mediated by the same antibody. Antibodies can also recruit other effector cells, for example, neutrophils, mast cells, and eosinophils, and other antibody effector mechanisms, for example, the generation of inflammatory cytokines or reactive oxygen species, like the equivalent of bleach. The advantage of having multiple effector functions in the same antibody is that they can add together, compounding the efficiency of killing. The disadvantage is that they can interfere with each other. For example, the generation of inflammatory cytokines and reactive oxygen species can lead to tumor cell killing, but they may also generate an immunosuppressive tumor microenvironment, which promotes tumor progression. And NK cells are particularly sensitive to reactive oxygen species. So it's a bit of a balance. Given the possible biologies triggered or blocked by the binding of an antibody to its target and the multiplicity of immune effector mechanisms, it can be very difficult to be sure of the main mechanisms by which an antibody achieves its therapeutic effect. But perhaps it doesn't matter if it works. Now, the first attempts to develop antibodies as pharmaceuticals started in the 1890s with horse and cattle antiserum, but they were seen as foreign by the human immune system, leading to what was termed serum sickness. There was another hurdle. Typically, an immune response generates many different antibodies to each antigen, often to different regions or epitopes, and with a spectrum of different activities that also differ from one immunized animal to another. For the development of antibodies as pharmaceutical products, it was necessary to find a way to create a single species of antibodies with a defined activity. And in 1975, Köhler and Milstein at the MRC Laboratory of Molecular Biology, in fact, Cesar Milstein was my boss at one point, they discovered how to make such monoclonal antibodies in mice by immunizing mice and immortalizing antibody-producing cells by fusion with a myeloma cell line. These hybrid cells secreted mouse monoclonal antibodies and could be grown in large fermenters for industrial production. But as might have been expected, the mouse antibodies were immunogenic in patients. That meant they produced an immune response that blocked the therapeutic activity and in some cases could be endangering of life. Fortunately, the advent of recombinant DNA technology allowed the engineering of more human forms of mouse monoclonal antibodies that were less immunogenic. First came the mouse-human chimeric antibodies, and then humanized antibodies, which we developed, as marked on the slide. Now, on the right-hand side, there's a white square, and that illustrates the architecture of the antigen-binding domains of an antibody. The associated heavy and light chain variable domains form a scaffold of beta sheet surmounted by 6 antigen-binding loops, 3 from each domain. To make humanized antibodies, we transplanted those antigen-binding loops from a mouse antibody in red to a human antibody in white by genetic engineering. And these antibodies, which are towards the bottom white with red tips, can be up to 95% human and with similar antigen-binding activities to the original mouse antibody. And unlike the mouse antibodies, these humanized antibodies were well tolerated in patients, and there are several antibodies of this class with blockbuster sales, including number 1, Keytruda. Now, the use of immunized mice to generate the antigen binding sites has the great advantage that mice can make antibodies against human proteins. By contrast, it's difficult to make human antibodies against human proteins due to tolerance mechanisms. We're hardwired to ensure we don't make antibodies against ourselves. Sometimes we do, and then we get an autoimmune disease. And in fact, if we tried to get those out of humans, actually, we've only usually discovered them because they're pathogenic, so it wouldn't be a good idea to use them in any case. Nevertheless, we thought there might be a way of making fully human monoclonal antibodies against human self-antigens. But we couldn't use immunization, so we had to think of some other way. And to understand our strategy, it helps to understand how the immune system makes antibodies against the entire universe of pathogens, many of which it's not previously encountered. Now, antibodies are made during the development of B cells. They're assembled from multiple gene segments by DNA rearrangements and editing. This happens independently in different B cells, generating a population of B cells, each cell encoding an antibody with a different structure and therefore potentially different antigen binding activity expressed on its surface. In a large population of B cells, there's likely a B cell that encodes an antibody cognate to any antigen. So it's analogous to a master thief who carries a large bunch of different keys, and if the bunch is large and diverse enough, he should be able to open any lock. So we aim to mimic this strategy. Firstly, we decided to create a large library of human antibody genes. And for example, if you look on the left-hand side of the slide, if we take 2 human B cells, so they're the red and the green cells, and copy the antibody variable region genes by polymerase chain reaction, we can expect to get 2 heavy chain genes, that's red and green, and 2 light chain genes, that's white and black. If we now link these genes together at random by the polymerase chain reaction, we can create 4 different heavy light chain combinations. 2 of the combinations will encode the original antibodies, that's red, white, and green, black, but the other 2 will encode new antibodies, that's red, black, and green, white, with new antigen binding activities that were not present in the original antibodies. Now, if we extend that example to 1,000 different B cells, for example, the number of plasma cells in 1 ml of blood, we could, for example, make 1 million different antibodies, of which 999,000 would be new. This antibody library will therefore contain or encode antibody binding activities that are not present in the original population, including those against human proteins to which the immune system is naturally tolerant. What's more, as both antibody heavy and light chains are entirely derived from existing human antibodies, these antibodies are fully human. That was the strategy. Now, clearly, these libraries of antibody genes could provide a huge and diverse library of antibodies, but how can we find the antibodies we want? I mean, screening 1 million antibodies isn't straightforward. And the problem is like finding a needle in a haystack. We needed a simple and rapid screening method. And our choice was inspired by the work of George Smith, who developed the idea of screening libraries of short peptides by displaying them on the surface of filamentous bacteriophage. Instead of peptides, we displayed antibodies. Unfortunately, when we cloned the antibody genes into the phage coat protein gene, they produced folded antibodies on the surface of phage, and they didn't abrogate the infectivity of the phage. So the phage were infective, and they displayed antibodies. We could capture the phage with binding activities to an antigen on a solid phase coated with the target antigen, and then we could selectively elute it, usually with a high or low pH buffer. The antibody genes can then be harvested from the selected phage and rebuilt into complete antibodies by, for example, for expression in a mammalian system. So we eventually use it as a way of fishing out the genes we want. And that's the process in essence, but I've missed out quite a lot of detail there. Now, with this technology, the same antibody library, the same tube of bacteriophage, so we're just about a small little tube of bacteriophage, could be used to isolate antibodies against one target after another. It can be regarded in a way as a bottled immune system, at least for the purposes of generating antibodies. And it was this technology that led to Humira, the first human therapeutic antibody to be approved by the US FDA, and for many years, the world's top-selling pharmaceutical drugs. It's only dropped now down to number 3 because of the generics or the biosimilars that have come onto the market. Now, at this point, it's worth pointing out the advantages and disadvantages of antibodies as therapeutic agents, comparing, for example, with chemical drugs. Antibodies have several advantages that here marked in the table as a green plus. Firstly, they can be derived by powerful selection processes, whether that's by immunization or phage display and recombinant DNA technology. This is quicker than de novo chemical synthesis and screening libraries of chemical compounds. Furthermore, in contrast to small chemical drugs, antibodies are large and can engage with their targets over large and even flat surfaces, leading to tight and highly specific binding, and the ability to block the binding of protein ligands to protein receptors. Small molecules tend to get squeezed out between interfaces of proteins. As already mentioned, they have a much longer serum half-life and can recruit a variety of effector functions to kill target cells, so getting more bang for the buck, really, with an antibody compared to a small molecule. Now, in contrast, chemical drugs often need to have enough hydrophobicity to bind to protein pockets, to cross membranes, and to be retained in circulation by binding to albumin. And this means that quite often they bind to other proteins that happen to have hydrophobic pockets, and that leads to off-target toxicity. Of course, you can have on-target toxicity, where actually you've got a therapeutic action for antibody. It can be toxic, but the real problem with small molecule drugs is they give you completely unknown toxicities that you wouldn't predict from their known target. Now, so I've given a list of great advantages of antibodies, But they do have some disadvantages, and here they're marked in the table as a red minus. In contrast to chemical drugs, they cross slowly from blood vessels into tissues. They don't enter cells except via pinocytosis or receptor-mediated internalization. And they have to be injected into patients rather than giving orally, and they have to be manufactured by cell culture rather than chemical synthesis, which increases the cost of goods and time taken for scaling up production, and actually means the regulatory processes are much more complex. They're also potentially immunogenic. Even humanized and human antibodies, given repeatedly, can generate human anti-idiotypic antibodies which block the therapeutic activity. But actually, it's possible by genetic engineering to improve antibodies a bit further. This is easier than making chemical iterations on chemical drugs. And it also helps that the multi-domain structure of antibodies tolerates a lot of changes. And I've marked possible improvements in blue in the table. For example, it's possible to extend the serum half-life of human antibodies from 1 to 2 months by making mutations that improve binding to the recycling receptor. Likewise, antibodies can be rendered more potent in killing target cells by enhancing their binding to the receptors on NK cells. As well as tuning the existing effector functions, it's even possible to build in entirely new functions, for example, by creating bispecific antibodies that activate cytotoxic T cells when they make contact with tumor cells. Furthermore, the hinge region of the antibody is amenable to chemical modification, and highly potent cytotoxic drugs can be conjugated to this region. And these antibody conjugates can kill cells directly if they're internalized by cancer cell receptors. So a different mode of action from having to recruit other immune cells in. Now, notwithstanding the impressive list of advantages, antibodies still suffer from some significant disadvantages. In particular, they cannot be made by chemical synthesis, they're large, they don't penetrate rapidly into tissues, and they're potentially immunogenic. Which raises the question, in our minds, of whether it would be possible to create small antibody mimics that combine the best features of antibodies with those of chemical drugs. So let's start off with the antigen binding site, marked here on the left. It consists of a large protein scaffold which supports 6 peptide loops of hypervariable sequence. But do we really need so many loops or such a large protein scaffold? We wondered if we could get away with 2 loops and a tiny chemical scaffold. We used a simple design in which a peptide with 3 cysteines was linked to a chemically reactive scaffold through cysteines, that's shown on the right-hand side, to create a bicyclic peptide. Large and diverse peptide libraries could then be created by varying the amino acid sequences, the locations of the cysteines, and the nature of the scaffold. So we can make different loop sizes, we can vary the sequences in there just using recombinant DNA to encode those regions. Now, in order to find those peptides with antigen binding activities, we use synthetic DNA to display the peptide libraries on the tips of filamentous phage, and we first made phage libraries of linear peptides and then added the chemically reactive cross-linking reagent to the phage. And again, I've missed out a lot of detail. If you're in danger of cross-linking all the phage, if you do it wrong, but you can just get it right and succeed. And to cut a long story short, Christian Heines, now professor in the EPFL in Lausanne, when he was working with me, selected a library against targets of interest, such as receptors, ligands, and enzymes, and he could isolate phage with binding activities, and from the selected sequences, chemically synthesize the corresponding soluble bicyclic peptides. So if we're going to use the therapeutic, we obviously need to take them off the phage and resynthesize them. Now, I've summarized the properties of these bicycles on this slide, and going down the list from the top, the soluble bicycles are small, typically 10 to 15 amino acids, and they penetrate deep into tissues. Nevertheless, they're large enough to have potent and specific binding affinities to their targets in the sub-nanomolar region. Like antibodies, bicycles have a scaffold structure, but the chemical scaffold of bicycles is more stable as it's fashioned entirely from covalent bonds. Not only are bicycles stable to heat, but they're relatively resistant to proteolysis. Synthetic bicycles can be made even more resistant by making substitutions in the loops with non-natural amino acids. Now, although the bicycle loop regions are more constrained than in a linear peptide, in fact, that's the reason we use them, because that extra constraint allows you to achieve higher binding affinities than you would with a linear peptide, which are very floppy. These are nevertheless floppier than the loops of proteins, and they can form a range of structures, including loops, hairpins, and helices. But given their small size and given the stellate core, it seems unlikely that they would be a prime candidate for loading into MHC molecules for presentation to T-cell receptors, even if partially digested. Now, I say that cautiously because we have the world's expert on T-cell receptors interactions sitting there in the audience, so I'm slightly alarmed he might contradict me. But nevertheless, whether or not he contradicts me, we've not discovered any immunogenicity from these small mimics. I wouldn't have expected them to be immunogenic, and simply in mice or patients, we don't see anything. Now, like antibody variable domains, bicycles are tolerant of other entities chemically linked to their C-termini. And this reflects the fact they were selected while anchored to the phage at their C-termini. So actually, previously, they worked with a huge phage attached to their end. So in fact, you can pretty much stuff anything else on the end of a bicycle, and it's still going to work. So you can append other entities, such as chemical toxins, or in fact, you can even stick bicycles together to create multimers, which give you an increased avidity of binding, or you can introduce biological effector functions. And I think it's fair to regard these multifunctional bicycles, in some respects, as tiny chemical mimics of antibodies, but with some other properties that may be advantageous. So the company Bicycle Therapeutics and its collaborators have shown that bicycles can be used to mimic the actions of antibody pharmaceuticals in several respects. A, as blocking antivirals. Sorry, I should have moved on. Sorry, how do I go back? Go back there. Yes, so blocking antivirals. For cell killing by recruiting immune effector functions, and for cell killing by use of toxin conjugates. I don't have time to go into this very much, but this just shows the principle, but also points out some differences with antibodies. For example, as I mentioned earlier with antibodies, there are multiple immune effector functions crammed together in that stem. With bicycles, we can dial in the effector function as we require and make them much more precision molecule. For example, we can take a bicycle against a tumor target, such as Nectin-4, and couple it to a bicycle against a receptor on an effector cell, such as NKp46 on NK cells, and we can use that to target NK cells to the tumor. Or, for that matter, CD137 on T cells to activate and target cytotoxic T cells to the tumor. And Bicycle Therapeutics has shown that such constructs can destroy tumors in animal models, and certainly the Nectin CD137 bicycle is already in phase 1 clinical trials in humans, and it's looking quite promising. Now, finally, I'd like to turn to the bicycle toxin conjugates. This slide shows one example used to deliver the chemical toxin auristatin to cancer cells. The toxin was linked to the bicycle through a spacer and a cleavage site for a protease. Unlike antibodies, the conjugates can rapidly escape the blood to penetrate solid tumors. The conjugate's not immediately toxic, as the hydrophilic peptide moiety prevents the conjugate penetrating the cell membrane. But when the conjugate is cleaved by proteases, which, to my surprise, are swimming all around tumors, the toxin is released and enters and kills cells bearing the target antigen, and also others throughout the tumor. So you've kind of got a toxic blast in the vicinity of the tumor, which in fact will also kill the adjacent cells that may perhaps have lost the cancer marker. And the excess uncleaved peptides cleared through the kidneys and disappears quickly from circulation. Now, this shows bicycle toxin conjugates in a mouse. And this was, what we did here was to destroy a large mass of tumor in a mouse. In this case, the target antigen was the metalloprotease MT1. And the experiment starts with mice bearing large tumors, a volume of about 1,000 millimeter cubed. In the vehicle control, black, the tumor doubles by day 14, and the animals have to be euthanized. But the addition of the toxin conjugate, a dose of 10 mg per kilogram biweekly, leads to the complete response, which is the green line after 28 days, with the only trace of the tumor represented by fibrotic tissue. Now, such preclinical data encouraged Bicycle Therapeutics to enter human clinical trials. And there are now 3 bicycle toxin conjugates in trials. One of these conjugates, the Nectin-4 bicycle toxin conjugate, has shown promising results in phase 1 trials for metastatic urothelial cancer. 6 patients have responded with 1 complete remission and a median duration of response of more than 11 months. So really very, very interesting results for that tumor. So now we come to the final slide. In summary, we think we can create effective antibody mimics using the Bicycle platform. And here I've compared the properties of bicycles, of engineered antibodies, of chemical drugs, and bicycles at the end. Going very briefly down the table, I've described to you how, like antibodies, they can be derived directly by phage display, how they can have highly specific and potent binding activities comparable to antibodies. Unlike many chemical drugs, they can block protein-protein interactions, and what I've not shown you is their serum half-lives can be more readily extended, for example, by addition of C-terminal fatty acid moieties. Now, like antibodies, bicycles can be engineered to bind 2 targets at the same time and mediate tumor cell killing and antibody effector mechanisms, or T-cell effector mechanisms, or via chemical toxins. Like small chemicals, their small size means they can escape the vasculature and penetrate solid tumors. But we've not yet engineered them to penetrate cells passively or to be delivered by the oral route. And I think those will be challenges. So currently, bicycles can mimic antibodies in multiple respects and can combine some of the advantages of small chemicals with some of the advantages of antibodies. And there will be some areas where antibodies are always better. For example, I think in the sustained blockade of protein ligands and their receptors, that's likely to be an area that would be difficult to beat an antibody for long-term maintenance in circulation. There may be other areas where they'll have advantages over antibodies, for example, as toxin conjugates, where a nice, simple, clean molecule able to penetrate fully into solid tumors, and also where you want to activate very specific immune effector functions and not have to try to remove them from your antibodies. There are also other areas where neither antibodies or bicycles have yet to establish a role, and in which bicycles may be better suited. For example, bicycles offer a source of small high-affinity synthetic tags for delivery of radiopharmaceuticals, nucleic acids, viruses, nanoparticles, or even existing chemical drugs to cells or to tissues. A small peptide as an address seems much more desirable than attaching a huge protein. Now, more generally, bicycles may be an early example of a new wave of medicines, a new paradigm shift in which synthetic chemistry is being used to mimic biologicals. And for this purpose, they seem well suited as building blocks to replace proteins. You've already seen another example from the mRNA nanoparticles of BioNTech and Moderna vaccines. They're, in essence, virus mimics made entirely by synthetic chemistry. And I suspect we're going to see, if we're thinking about new paradigms in biology, this may be one. I think we'll see a lot more examples of this kind of thing in the future, limited only really by our imagination. Thank you.
Thank you. Thank you so much, Professor Winter. Please be seated. Thank you so much for your insightful sharing.